At first glance, the cardiovascular system may seem far too complex to study in a tiny chip. It spans the entire human body, with pulsating arteries, capillaries thinner than a hair, and intricate biochemical interactions. Yet advances in microfluidics, the science of manipulating small volumes of fluid within microscale channels, have made it possible to recreate and study key aspects of the cardiovascular system on platforms smaller than a credit card. These organ-on-chip and vessel-on-chip technologies offer a new frontier for biomedical science. Precise, controllable, and physiologically relevant models of heart and blood vessel function. Why use microfluidics to study cardiovascular systems? Traditional cell cultures are flat and static, failing to replicate the dynamic 3D environment of living tissues. Animal models, while more complex, often differ significantly from human physiology and raise ethical concerns. Microfluidic systems bridge the gap by offering physiologically relevant environments in a highly controlled format. They allow researchers to simulate blood flow, pressure, and shear stress, and observe how cells respond in real time. This level of precision opens new doors to understanding disease mechanisms, testing drugs, and even tailoring treatments to individual patients. How does it work? A microfluidic chip typically consists of polydimethylsiloxane (PDMS) channels bonded to a glass substrate with embedded chambers mimicking blood vessels or heart tissues. These channels can be lined with endothelial cells, the same cells that coat human arteries and veins. By pumping fluids through the system at specific rates, researchers can reproduce physiological flow patterns and mechanical forces. When these cells are subjected to pulsatile flow or cyclic strain, they begin to exhibit behaviors remarkably similar to their in vivo counterparts, including alignment, nitric oxide production, and inflammatory responses. Consider a microfluidic model of atherosclerosis, the disease in which plaques build up in arteries. Scientists can introduce cholesterol-rich particles into a vessel on chip lined with human endothelial cells. Over time, the cells begin to show dysfunction, mirroring the early stages of cardiovascular disease. By precisely controlling the flow rate, pressure, and biochemical environment, researchers can dissect how mechanical stress and metabolic cues converge to cause damage. Insights that are difficult to capture in living organisms. Another breakthrough lies in heart-on-chip devices. These systems use microchambers seeded with cardiomyocytes, heart muscle cells derived from stem cells. By integrating electrodes and flexible membranes, these chips can monitor contraction force, electrical activity, and response to drugs. In one example, heart-on-chip models have been used to predict the cardiotoxicity of cancer drugs before human trials, offering a powerful new tool for drug screening and safety assessment. This capability becomes especially transformative when paired with patient DNA cells, enabling personalized testing and even predicting individual risk of adverse reactions. What about more advanced features like blood clots or immune cell circulation? Microfluidic platforms can model these too. By incorporating microvalves, branching channels, and 3D printed scaffolds, researchers can simulate thrombosis, clot formation, leukocyte adhesion, and platelet activation under flow. These systems allow for high-resolution time-lapse imaging and can track the cascade of events from inflammation to occlusion. With these insights, scientists can evaluate antithrombotic therapies under realistic conditions. Which fields are benefiting from this technology? The impact spans cardiology, pharmacology, biophysics, and tissue engineering. Pharmaceutical companies use cardiovascular chips to reduce reliance on animal testing and accelerate preclinical trials. Academic researchers explore the cellular basis of diseases like hypertension, aneurysm, and myocardial infarction. Engineers use microfluidics to study how mechanical forces regulate gene expression in vascular tissues, a critical area for developing better stents and vascular grafts. Emerging tools are taking this field even further. Multi-organ chips connect cardiovascular models with liver, kidney, or lung chips to observe systemic drug effects. Artificial intelligence is being applied to analyze large data sets from microfluidic experiments, identifying subtle patterns in cell behavior. 3D bioprinting now enables the construction of anatomically accurate, vessel networks within microfluidic devices. These hybrid technologies signal a future where personalized, high-throughput, and physiologically accurate testing becomes the norm. Ultimately, microfluidic tools are not just miniaturized labs, they are living models of the human body. By shrinking biology onto a chip, researchers gain unprecedented access to the forces, flows, and signals that govern cardiovascular health. Could these devices one day replace animal models, personalize medicine, or predict heart attacks before they occur? The growing convergence of microengineering, stem cell biology, and data science suggests they just might. Microfluidic devices precisely manipulate microliter volumes of fluid through networks of channels whose dimensions mimic the scale of human blood vessels. This scale enables accurate replication of hemodynamic forces, shear stress, pressure gradients, pulsatile flow that govern endothelial cell behavior in vivo. Integrated sensors within these chips allow real-time measurement of cellular responses, biochemical secretions, and mechanical properties under controlled conditions. By bridging the gap between traditional cell culture and animal models, microfluidics delivers physiologically relevant data with high throughput and reduced reagent consumption. Example: a straight microchannel lined with endothelial cells reproduces arterial shear stress profiles to study barrier function under flow. Analogy: a microfluidic chip is like a miniature plumbing system that recreates blood flow dynamics in a petri dish. Shear stress, the tangential force of fluid flow on vessel walls, is a critical regulator of endothelial gene expression, morphology, and function. Microfluidic platforms allow precise tuning of shear magnitude, direction, and pulsatility to mimic arterial, venous, or disturbed flow patterns found at vascular bifurcations. This controlled environment reveals how different shear regimes drive protective versus pathological endothelial phenotypes, informing mechanisms of vascular health and disease. Quantifying cell alignment, nitric oxide production, and inflammatory signaling under defined shear helps identify therapeutic targets for vascular dysfunction. Example: endothelial cells in a bifurcating microchannel develop pro-oscillatory markers under low oscillatory shear, replicating atherosclerosis-prone regions. Analogy: it's like testing how plants bend differently in a gentle breeze versus gusty wind by adjusting airflow in a wind tunnel. Vessel-on-chip devices recreate three-dimensional tubular architectures lined with endothelial cells and surrounded by supporting extracellular matrix and perivascular cells. These biomimetic vessels permit study of cell-cell and cell matrix interactions under realistic geometry and flow conditions, capturing barrier function, permeability, and angiogenesis. Incorporating smooth muscle cells into concentric layers enables investigation of vascular tone and remodeling in response to biochemical cues. Such chips provide scalable platforms for screening drugs that modulate vessel integrity and function. Example: a cylindrical microchannel embedded in collagen demonstrates how VEGF gradients induce capillary sprouting. Analogy: a vessel on chip is like a tiny garden hose embedded in soil that sprouts new branches when watered with growth factors. Microfluidic shear and stretch actuators simulate cyclic strain experienced by endothelial cells during cardiac pulsation, influencing cytoskeletal alignment and gene expression. Controlled mechanical stimulation reveals mechanotransduction pathways that convert physical forces into biochemical signals, regulating inflammation, thrombosis, and vascular remodeling. By isolating individual mechanical parameters, researchers dissect how aberrant forces contribute to diseases such as hypertension and aneurysm formation. This insight guides design of mechanotherapeutics targeting force signaling molecules. Example: applying 10% cyclic stretch in a microfluidic chamber upregulates endothelial nitric oxide synthase, mimicking exercise-induced vasodilation. Analogy: it's like repeatedly stretching a rubber band to study how tension influences its elasticity and eventual wear. Microfluidic blood flow assays recreate vessel injury by introducing collagen or tissue factor surfaces under flow, triggering platelet adhesion, aggregation, and clot formation. High-resolution imaging of clot growth dynamics reveals real-time interactions between platelets, coagulation factors, and fibrin networks under physiological shear. These models allow evaluation of antithrombotic drugs in human whole blood without animal testing and with precise control of hemodynamic parameters. They also help identify patient-specific hypercoagulable states for personalized therapy. Example: a stenosis mimicking microchannel under high shear shows accelerated thrombus formation that is attenuated by a novel antiplatelet agent. Analogy: it's like observing how leaves pile up behind a rock in a stream to understand how blockages form and can be cleared. Microfluidic organ-on-chip platforms integrate vascular flow with tissue compartments to study drug absorption, distribution, metabolism, and toxicity under dynamic conditions. Continuous perfusion mimics blood clearance, enabling accurate measurement of drug half-life and off-target effects on endothelial integrity. Parallelized chip arrays facilitate high-throughput screening of candidate compounds, accelerating lead optimization while reducing reagent cost. These systems better predict human responses compared to static cultures or animal models, improving translational success. Example: a liver vascular chip quantifies cardiovascular toxicity of a chemotherapy agent by monitoring endothelial barrier leakage under flow. Analogy: it's like testing a car part on a moving conveyor belt rather than stationary bench test to see how it performs in real driving conditions. Complex vessel-on-chip systems replicate plaque development by co-culturing endothelial cells, macrophages, and smooth muscle cells under pre-atherogenic flow and lipid exposure. Chips enable visualization of lipid accumulation, foam cell formation, and extracellular matrix remodeling in situ. Controlled perturbations, e.g. hyperlipidemia, inflammation, reveal disease progression steps, and allow testing of plaque-stabilizing therapies. This approach provides mechanistic insight and therapeutic screening in a human-relevant context. Example: exposure to oxidized LBL in a bifurcated microchannel induces localized foam cell deposition, mimicking early plaque. Analogy: it's like growing rust patches in a miniature pipe under accelerated corrosion conditions to study blockage formation. Microfluidic chips, seeded with endothelial cells derived from patient iPSCs, capture individual genetic and epigenetic variations in vascular responses. Personalized chips reveal patient-specific drug sensitivities, risk of thrombosis, and endothelial dysfunction, guiding tailored therapies. Longitudinal chips track disease progression and treatment response over time without invasive procedures. This individualized approach promises precision cardiovascular medicine based on functional assays rather than population averages. Example: iPSC direct endothelial cells from a diabetic patient show heightened permeability under high glucose flow, reversed by targeted therapy. Analogy: personalized chips are like custom-fitted shoes. They match the unique contours and needs of each individual. Next-generation platforms combine vascular, cardiac muscle, and immune cell compartments with embedded sensors to model whole organ physiology on a chip. Integrated flow control and real-time analytics enable simulation of systemic responses to drugs, stress, and disease in a single device. Advances in 3D bioprinting and microfluidic automation promise fully automated, multi-organ networks for comprehensive pharmacological testing. These holistic hard-on-chip systems aim to revolutionize cardiovascular research by providing human-relevant, scalable, and predictive models. Example: a multi-organ chip links cardiac tissue beating under physiological load to a vascular network, monitoring drug-induced arrhythmias and endothelial injury simultaneously. Analogy: integrated hard-on-chip is like a miniature city model where roads, buildings, and utilities interact dynamically to reveal how changes in one district affect the entire system.